Hello and good evening to everyone who has joined us live today and to those who will be watching this recording a bit later. I am Udovč, a journalist at the Slovenian press agency, and I'm really honored to welcome here with me Laura Boschis, a chemist and a CEO who started her career path in telecommunications industry, going then into academia and later on launching a startup company called Trustech, of which Laura is now CEO. The title of today's talk is Lab on Chip and Point of Care, from idea to productions, which means we will hear more about the path necessary to design a lab on chip and the difficulties that must be faced to achieve industrialization phase. I will ask everyone to mute themselves if possible, and if you have any questions during the talk, please write your question in the chat. If not, you can wait until the end of our talk and also write the questions in the chat, or just raise your virtual hand and we will give you the word. So Laura, your career path is quite unusual and very exciting. Can you tell us more about how did a chemist like you come from telecommunications to biology and to developing micro device that can, among other things, make a huge impact on the future of personalized medicine?
Okay, thank you, Leah. Okay, just to introduce a little bit of my story. Now I'm the CEO of Trustech, that is located in the north of Italy, close to Torino. And here are my references, my numbers, my email, and everybody is free to send me email later. Next week. What I began I want to say a little bit of myself, but not because I'm so important, but just to tell a story of what happens to a lot of people. I studied chemistry, and when I was young, I was really fascinated from organic chemistry, and I thought for a long time to become to begin to work in the synthesis of molecules, organic molecules. But even if I studied chemistry, actually I never worked as a chemist in the most iconic idea of chemistry work. In fact, I was for the first 10 years of my life, 10 or 15 years of my working life, I was involved in the electronic and optoelectronic device technologies. But when I was a chemist, I really thought to become. When I was studying chemistry in the university, I really wanted to become an organic chemistry. But at the end of my studying, I end up to work in the telecommunication centers, Italian telecommunication centers, and then I begin to approach semiconductors, but in particular 3-5 semiconductors that are essentially indium phosphide, gallium arsenide, and most people has no idea, even if most people has no idea what they are. Without this kind of materials, all of our devices will not work because they are important in the side of the devices, but especially they are very important to send data in optical fibers, because they are able to emit, this kind of semiconductors are able to emit exactly in the narrow windows in which glass of optical fibers guide. And so I begin to work with materials that are very ordinated, crystalline materials. I liked them a lot because they are precise, well organized, educated and disciplined as well. But after some time, I moved and I began to work in the devices directly in optical fibers. So my working life, my working subjects was a little bit different. Then I moved from crystal to amorphous. And I began to work and dealt devices inside of the fibers. So glass that is amorphous, undisciplined, that is anarchic in a certain way, was my new jobs. And so I acquired a little bit of experience even in other kind of materials, other kind of subjects. In that period, the world, I'm talking about the 90s, was really crazy because all people, everyone wanted to join new records in the sense of devices performances, because there was the idea that internet has to go inside of every home, everywhere. So there was a so high rush to obtain the best performance of devices, because megabytes were not enough, then gigabytes, then terabytes, and every day there was somebody that tell, oh, your device is not so performing, we need more and more. But at a certain moment, everything was stopped. It was clear that it was a bubble, and a real speculative bubble. So after a big period of euphoria, where everybody was able to pay optical engineering a lot of enterprises immediately closed their doors. Investors were not available to find more research, and a lot of people like me lost the work. Lost their work. And they had to invent themselves again, because what can do in the world a person that is specialized on 3-5 semiconductors or optical fibers, if not working on telecoms? But we had to think. We had to think a lot of our lives, or what we acquired, what experience we acquired, and somebody, because the crisis let your wheels inside of the brain and move faster, realized that the high miniaturization that we obtained was compatible with the big molecules of biologists. So it was really possible to use our technology, the technology of for silicon or for other semiconductors. Not only there was a lot of other not only gallium phosphide or indium phosphide, but even gallium nitride. A lot of others are possible to be employed in the detection of molecules, for example, big molecules. It was possible to measure them in a new way, in a new concept way. And so instead of let our tears on the rain, we begin to think new devices. New devices like, for example, lab on chip. In that period, I moved from the company that I worked in the Agilent Technologies company for 5 years. And then when they decided to close the activity, the research activity on telecom, I moved in the Polytechnic of Torino, that is a research center, a university close to me. And there, for the first time, I faced biologists, because I went there with the mission, with the object to realize, to employ the specialization that I had on semiconductors directly in the biology field. And so we faced the first problem. The first problem is that the engineer had to talk with the biologist, and biologists speak not our languages. So we had to find a ground zero in which we had to talk together. Medicine and biologists really didn't understand technological aspect, physical, chemical, technological aspect, and vice versa, the engineer or a technician were not able to understand them. So a ground zero. For some years, we try to understand, to find how we can cooperate, which kind of devices could be possible to realize. We understood that it was possible, but how? And in that period, the lab on chip were burnt. What is a lab on chip? A lab on chip is a small devices in which it is possible to produce or to analyze very small quantity of solution in a small surface, few square centimeters. In few square centimeters, it's possible to reproduce both analytical protocols and detection that could be realized with an electrical that should be electrically performed, or mechanical, measure mechanical structural stress, or at least optical one. So these new devices had the problem of to find finance, how to find money to perform, to manage our research. And the problem was that the classical funding platform were not at the moment still able to understand, ready to understand the new technologies. So when we tried for the first times to write a proposal, both in Europe and both in our local, regional or national platforms, if we try to present a proposal in a platform for biologists, they say, oh, but your device is not so unusual. In biologists, PCR is a normal protocols. We are already ready to do that. We know that it is not so innovative. But if we put our devices in an engineering platform, they say, okay, your device is not so performing. You are doing something that it is not so innovative from the point of view of the device. But the innovation was the junction between biology and engineering. So a lot of people in the university tried to create for this reason startups with the idea of funding money with bank, with incubators and so on. But startups in these fields so new, in which there was not a ready market, a lot of them failed because they were too much fragile. There are a lot of problems to get money, to attract investors, to attract expertises and so on. But in that period, that was around the 2008, more or less, me and other people, other partners, decided to hurt ourselves and to create pain by ourselves. So because the worst enemy that we have sometimes are ourselves. So we create a startup, Trustech as well, that is my company, that had a lot of problems. It sailed always in a very stormy ocean, but we are still alive, still fighting, and we are here to continue to our research, to continue to perform our research, paying our own research from one side with the public funding, of course, and from another side with the solving problems of our customers, technological problems, of course. So Trustech is...
Laura,
yeah?
Sorry, Laura, could you give us just a very brief and short idea of what Trustech does today? What is the general idea of the company? Yeah. What do you do?
We are an innovative SME in which we perform R&D, both for our customers, both for our service, for internal commitments. And in our commitments, we produce by our service analytical platform. We are focused on sensors, especially sensors, chemical sensors, and in particular biochemical sensors, and in particular lab on chip. So we perform MEMS, that are microelectronic mechanical systems. It is an acronym created after the new idea of how to apply semiconductor technologies, and from the other side, the lab on chips. And we are ready to speed in the industrialization, the first of our platform, right in these periods. So Trustech is a multidisciplinary company, a very small one, but with a lot of experience, and with people coming from different experiences that come together. Part of the personnel is here directly, but a lot of them are linked in a network, and part are working with us just when it is necessary. So what we did, we are together in order to solve biological problems, and the biological problems, trying to see in a wider vision, in the wider vision as possible. And what I wanted to recommend to everybody is sometimes to reach again the surface, and not go too much into the detail. That is very useful for the early years of the work experience, but then it is very important to research and see what you can see, what happens around you. So by snorkeling and not diving, you can have a wider vision of the world of the technologies, and you can begin to speak with the monads close to you. It is not true, as Leibniz said, that monads has no windows. We have to open the windows, open all our windows, and begin to speak with chemists, with biology, with engineer, and normal people in the street, just understand all the experience, and create new things, new ideas. And that is what happens for the creation of microfluidics. Microfluidics is a building blocks useful for lab on chip. Lab on chip is not only microfluidics, but microfluidics is something that is very useful to create a miniaturization lab, because in reagents may be moved through microchannels according to a certain well-designed pattern, moved around along through channels, arrive in reactive rooms, small rooms inside of this in any surface, and they are able to react together to produce some things. And at the end of the microfluidic, we can have channels, reservoirs, reactive chambers, and at the end, detection area, if we want. But to create that is necessary to talk with to join a lot of competencies.
Right. So the basic idea of the lab on chip is to miniaturize a complex laboratory processes into a principle to, in a way, speed up the processes and to reduce the amount of sample needed, if I understand correctly. And the technology of the laboratory is based on microfluidics, which is the science of the behavior of liquids in narrow channels. Right, Laura?
More or less, yes. More or less, yes. The problem of the idea of the miniaturization, be careful, is not to reach a record, to go in the world record as the smaller devices in the world. But the idea of the miniaturization is important, and miniaturization is important because it emphasizes the sensitivity of the device. For example, it is useful to enhance the surface volume ratio in a reaction in order to accelerate the kinetics. So reaction in lab on chip normally may be faster, much more effective, and rapid as well, and precise if everything is well designed. So the roadmap to obtain a lab on chip, for example, just to give you an idea, a microfluidic lab on chip has to be, first of all, to study the problem, as I told you before, and understand, because not in every situation it is useful to use microfluidics. But first of all, we have to say it is right to use to apply microfluidics for these single protocols, for these single problems, this peculiar problem. Yes or no? If yes, okay, we have to analyze which kind of steps we have to replicate in the surface, in the microfluidics, with which amount of reagents, which reagents are necessary, are they with which materials are them compatible? Is it necessary to store them for a long time? So in order to, because it is important to avoid permeation of the reagents inside of the materials, that is typical, more or less always typical, is a polymers. So not every polymers is suitable for every kind of reagents. So we have to understand, and at the end, which kind of detection we will use. It's an optical detection. It's an optical in which range of the spectra, visible, infrared, or UV. So we need a specific materials that is transparent or reflective in that peculiar area, in this peculiar range of emission or transmission, or we want to analyze the detection with an electrical way, with resonance or something like that. So we need something that in some place, in some way, and where we want is conductive, and otherwise not, because we have to insulate all the other area except the electrodes. So, and how can we do our electrodes? We evaporate them, we put some contact points inside. All these problems is to be analyzed, organized, and then we have to design the whole projects. And then another important aspect that sometimes researchers don't have in mind is, at the end, which will be the amount of the production. That means if we will produce 100 devices per year, we can use certain technologies. But if we say 1 million devices per year, probably we have to choose something that is reproducible in a great amounts, that is low cost, that has a low cost, and it should be compatible with industrial protocols, industrial production steps, and so on. So it is very important even to talk at the very beginning with expertise of industrialization. That is not so common inside of the university, but this is one of an important aspect. Because at the end, if not, you risk to arrive to a very good result, good for papers, good for research, good for what you want, but at the end, it will be not a product. The step from project to product is a hard way that is called, first of all, that passes through two steps: in the industrialization engineerization and industrialization that are very difficult steps. Usually university people are not used to think in such a way, because I know, because at the very beginning I work in the university, so I know that this is a different path, and even because it is quite frustrating, because during the industrialization or engineerization, and worse, still worse, in the industrialization path, you don't publish anything, because you are replicate your test, you are replicate your results, because the problem is here is to demonstrate that your work is working a million of times always as well as the first one, that you have a good reproducibility, that the live test is going well, that reagents are maintained efficient and not oxidized for a long time. So it is not important, it is not an interesting value parameters to be published, but it is an important steps in order to arrive to a real product. And then
talking about industrialization, you already have one device that is almost ready for industrialization, right?
Yes, it is ELICHIP. ELICHIP, that is our platform in which we obtained a point of care method to perform immune enzymatic analysis everywhere. So it is very versatile, because it could be used in a lot of kind of fields, starting from food safety, arriving to veterinary application, arriving, and at the end in medical care. Medical care, that means close to the patient, at home, or in a medical cabinet, or in a laboratory in which there are some researchers that need a single value, single analysis, not a wall plates. ELICHIP is, in brief, is very easy, and it is possible to use ELICHIP when it will be completely performed as a product, as it will be usable, and it was so easy to use as a coffee machine with the caps inside. You can obtain quantitative immune enzymatic analysis with the same precision of gold standard in the laboratory in a single analysis. Inside of during the analysis, every time you do as not only the analysis, but even the calibration, titration curves, in order to have a very real quantitative analysis. And this is the reason, because, and just because we use a very small amount of volumes, it is possible to be used even applied to organ on chip. And this is the reason, because ELICHIP was chosen as one of the lab on chip for the FLAMIN-GO projects.
So let me say a few words about the FLAMIN-GO project. The general aim of this European project is, in a way, personalizing the treatments for rheumatoid arthritis, a long-term autoimmune disease that causes pain, swelling, and stiffness in the joints, and for which there is no certain cure yet. So the main technical goal of the project is to develop, as you said, an organ on chip. This is a very small cell culture unit that could be used to test whether and how a patient will respond to a specific drug before a drug is actually given to him. So this way, serious side effects of some drugs could be avoided and the treatment speeded up, thus improving patients' quality of life. And where does your lab on chip, Laura, the ELICHIP, come into story? How does it interact with the organ on chip? What is its role in the project?
From the organ on chip, the cells inside of the organ on chip are fed with vascularization, with a vascularization, and the outlet of this vascularization arrives directly could be directly analyzed in ELICHIP. For the moment, it was chosen by the partners of the project four molecules, two ketokine and two matrix metal proteinase, in order to understand, to follow the average, the concentration of these molecules, analyze the inflammatory, the level of the inflammatory inside of the cell patient. ELICHIP is useful both for the diagnosis of the arthritis, synovial arthritis, and both for the analysis of the treatments. You know, that means that when the cells will be treated, the evaluate will give us some information, thanks to ELICHIP, to the quality or the validity of the treatments. So, but I told you that ELICHIP is not only usable in FLAMIN-GO. These are the first four molecules in which we wanted to say, but what is important to say is that we use a few amount of volumes, and this is a real tricky solution for an organ-on-chip, because from organ-on-chip there are so a few numbers of cells, and the weight coming from the organ-on-chip is so small amount that with a normal analysis in laboratory it will be not possible to be analyzed, because they require hundreds of microliters. But by the contrary, with the ELICHIP we can measure 10 or 20 microliters per time. So it is really a great improvement in this sense. And then, okay, the application I already talked about, but we are working even in personal medicine, but in food safety as well, or in veterinary, some veterinary application has tested as well, for example, analysis of Leishmania virus, etc. But I want to leave you with just a provocation that I know, okay, I told you about the story and how many technologies there is behind a lab-on-chip, but how much do you think that the market is willing to spend for a lab-on-chip, for a single lab-on-chip? I don't know if anybody has an idea of how much people or market is available to invest on that. But I want just to show you, just looking around in internet, that there is people that is ready to spend 37 euro for a school-shaped toilet brush. And there is even somebody else that spent 40 euro for a neckerchief of noise dispenser. So, and there is even people that review them and gave them 5 stars in Amazon marketplace. But as well, if the value of a toilet brush is 37 euro, 40 euros as well, you want in your toilets, people is ready to invest just no more than 10 or 20 euros for a single analysis. That is crazy world in which we are. And so I thank you, and if there are some questions, don't hesitate to send me an email or ask now, and then I want to let you with our logo and our brands. Thank you, Leah, and everybody.
Thank you, Laura. I have a few more questions for you, and maybe in the meantime, someone will also ask some of their questions. I advise you to just write your question in the chat, or if you want, you can raise your virtual hand and we will give you the word. But Laura, can you tell me what are some of the bottlenecks in the FLAMIN-GO project at the moment? Do you still have some challenges to overcome? At what phase are you at the moment?
Okay, I can talk about my own work, of course, not all the other partners' work, but from my side now we are facing with the solution, because until now we tested the molecules in with the standard solution without any kind of interferences. So now we are trying for the first time with real cell culture solution coming from cell cultures, not directly from the lab on chip until now, but with the real matrices in which we are. We see that until we are analyzing just a single molecules, it works well, but we have to let the solution become always much more complex and close to the reality. And so the dark area now is to understand if there are a lot of interferences from the matrix or not, and if our system method is working well as well.
And in what time do you hope for ELIChip to enter into practice, and where do you hope to see it? Is it in the medical room? Is it in the homes of patients? Anywhere else?
Well, our sites inside of my enterprise, we are going to find funds now. So we applied and we are in the first round for the industrialization, and we think that in 12 months we will be ready to the market.
Okay, perfect. And do you think that lab on chips in general could ever completely replace laboratories, or we will still need real labs?
It depends from the application, of course. There are some applications that are more suitable than others, but sure, that will be, that's the road, of course, but they will not substitute themselves with the classical big high throughput analysis, because they are very suitable for single patient analysis, single samples. With when you have to analyze big amount of samples of the same for the same protocols, of course, the big laboratory machine are the best that we have now. They are very solid. They have very solid protocols, consolidated protocols. They are designed and tested since a lot of years with to go fast, reliable, etc., etc. But for single analysis, they are not good enough.
Perfect.
So lab on chip will empty a lack zone.
Okay, just one last question, a more general one, just to see how you imagine the future. What are some of the things that you look forward in the future in your research field? What is something that you look forward to or hope to achieve in the future?
To enlarge our methods, to reproduce other methods as well, because we have other projects. Now we are going to test. They are in a seed stage for the moment, but we have other ideas that are for the moment closed for the just to protect the IP, because they are not protected for the moment with patents, so I can't tell you nothing more, but we have other project there.